It's a great pleasure to be here. We're going to switch gears a little. I'd like to talk about large-scale inference and topological analysis of microbial interaction networks. So my name's Christian. I'm at the Simons Center for Data Analysis, and for those of you who don't know us, we are within the Simons Foundation on 5th Avenue and 21st Street. And the idea of the Simons Center for Data Analysis is to design algorithms for biology. Okay, so we are very interested in algorithmic design and computational methods that are useful in biology. If you want to know more, there's the annual report. And my research is embedded in what we may call microbial systems biology. So what we know since a couple of years is we can generate large amounts of microbial data, and I go through this in a bit. And what we want to understand is various things from these microbial abundance data. We want to estimate species abundances in different hosts, across different habitats. And what we focus on today is estimation of microbial species interactions. Okay. All right. So as many of you may know, the scale of our micro-universe is quite impressive. So if this is the number of human population, we are vastly outnumbered by microbiota. And number of bacteria and viruses on this planet vastly exceeds the number of stars in the universe. And since about 10 years or so, we can actually access this large amount of bacteria using large-scale sequencing efforts, such as the Earth Microbiome Project and the Human Microbiome Project that uses 16S RNA sequences to find out what's in all these different habitats that surround us. And just for you who don't know what large-scale 16S sequences is doing, is basically, so what you can do is you can take a sample site, say a human or a lake or a soil, and you do PCR amplify the 16S RNA gene, several regions of that, and this gives you basically a tag for each microbe that's in there that you can assign a taxonomy and something that we call operational taxonomic unit. Okay, so you can take your sample, and then you basically find out what is the relative abundance of different species in the different samples. So here, relative abundances identified from the different samples. Okay. So a typical data would look like this. So here you have the OTU, so all the different species, and here are all your different sample sites. And then you can do cool analysis, and we focus today on interaction networks among all these different OTUs, all the different species. All right, so let's look at one specific habitat. So we know since a couple of years that the gut harbors a lot of microbes, and they have a key influence on neurodevelopment, metabolic diseases in our body, autoimmune diseases, and there is also pathogenic competition, okay? And what we really want to understand is this system as a whole. But we don't know anything about the interactions among these microbes in the specific habitats, okay? And why ecology is very important in this is, for instance, Costello and co-worker put this forward. So imagine you have a system here of microbes in your gut, and then what would happen to the system if you, for instance, treat it with antibiotics, or in your mouth if you have oral hygiene? Then you could imagine that the landscape, the state landscape, switches from one to the other. Well, what is happening if you change your diet? How does this system change? Okay. And we don't know, we draw these networks, but we don't know these networks yet. And that's the purpose of our work. So when I talk about interactions in general, there can be many different types of interactions among microbes, as you know. There could be physical interactions, cell-cell, metabolite-mediated competition, and so on. But we don't have access to this on a larger scale, right? So we could draw some of these maps on a fine detail for certain subsystems, such as here, how different microbes in the oral cavity interact, but we don't know this in general. So from the 16S large-scale data, there were a couple of methods that were put forward in the past couple of years, and we contributed to this. So the idea is really learn interactions just from the 16S sequencing alone. Okay, what are the challenges for that? So we know the 16S sequencing data that we have are intrinsically compositional, so they are not uncorrelated with each other. And then the question is, how should we measure on a large-scale interaction? You could use correlation. What we do is conditional independence, or partial correlation. And then we are also in a regime which is underdetermined, so we have many more bacteria that we actually measure than sample points, just depicted in this type. So we want to infer something among all the species, but we have very few samples. So how we do this is using speakeasy. So Speakeasy learns parsimonious direct interactions from OTU abundance data. And the workflow is like this. So you have your OTU data, you pre-process in a specific way such that you take care of the compositional property of the data. Then you learn a sparse graphical model, which is basically based on the lasso. We do stability-based model selection. What you get out of the pipeline in the end is a dependency graph among all the OTUs and a regularized covariance matrix of the abundances. Okay? So our key hypothesis for that is actually that not all bacteria, for instance, in the gut interact with all others, but actually that there is a sparse network. Okay? That's the key thing, such that we can compute anything in the first place. And this obviously relates to parsimony. Okay, just to give you an idea why conditional independence is better than correlation under certain conditions is, imagine you measure the abundance of 4 species, that's the true interaction network. Here's the data over many, many samples. If you compute the correlation matrix, it would look like this. If you now thresholded this correlation matrix and say everything above a certain absolute value is an edge, you could not recover the original network. Okay? But if you now use the inverse of this, which is also called the partial correlation matrix, then you can read off from the non-zero entries of this matrix basically the interaction. So here you see that 3 interacts with all the others and only these values are non-zero. Okay, so we can do this on a large scale and induce sparsity by assuming that this matrix is kind of sparse. Okay, so how does this work? So for instance, if we apply this to the American gut, one of the large projects here in the US, these are 2 types of networks we infer. There are 2 different algorithms that underlie Speakeasy, here colored by family and order. Okay, and if we compare this to the competitive methods, the other methods, Spark and Creep, they infer similar networks, but they are much denser. Most probably there are a lot of false positives in there. And we can quantify that by saying, okay, if you have 2 sets of data, you learn one network here, how similar are these networks given the same data source, then the reproducibility is much better for Speakeasy. So here we measure distance among the different networks compared to the other methods. All right. If you want to know more, there is a PLOS-COMB bio paper that explains this and many more tests. All right. So now what we can do is we can do large-scale learning. We have this tool. What we did is we collected all available 16S data today. It's more than 300, went through some quality control, took them from all the different servers there are, and now we can actually do ecology. We can look at interaction networks across multiple habitats. So we inferred more than 5 million interaction networks, potential interactions among different species in more than 300 different networks. Okay. All right, so now we are at network thinking in biology. So we heard a lot about transcriptional regulatory networks and also recently about genome interaction networks. And the question really is, how do microbial interaction networks fit into this global network picture? And we can ask a lot of ecological questions. For instance, is the organization of the networks different dependent on the habitat? Are there features that are common to all networks? Can we come up with a co-evolutionary model that explains the emergence of these ecosystems? And can we find a generative network model in general? Okay. Okay. So when you just look at these networks and visualize them, they look, depending on the habitat, they look very, very different, or can look very, very different. And now the idea is, okay, can we actually infer some similarity among them and infer some global properties? So to compare these networks, we use the graphic correlation distance that has been put forward last year. We basically have an unbiased way of comparing network structure across different sizes. And now I can show you a global similarity map of all the ecosystems network we inferred so far. So what you see here is each dot is a network, and the size corresponds to the size of the network, and the label corresponds to the habitat where it came from. Okay? So here, for instance, we have soil, water, and built environment networks. Down here, for instance, we have oral networks, interaction networks that live in your mouth. And just a reminder, here are the American gut networks we inferred, and they look like this. Okay? And here, this really means that if you're close in this map, then your network organizations are really similar. So that's the first global picture. And obviously a fundamental question is, having these networks now, can we say something about the stability of these ecosystems? And this question is as old as evolutionary theory itself. So Darwin already said something about this. He said, well, if we have a large number of species in there, then this is usually ecologically more stable, given a fixed area. MacArthur, 100 years later, said actually, well, this is why we increase ecological function. And May in the 70s actually said, okay, so but if we have more and more species given a fixed area, we should actually observe something like a decreased number of interactions among these species, such that the system is stable. And to put this in precise notion, this May-Wigner stability criterion is actually, so the percentage of edges you have in your interaction network should go down as 1 over d, where d is the number of species, OTUs, you have. Okay, what do we observe? We observe this. So if you look at the best fits, so theory predicts that this exponent here is -1. A best fit, however you fit it, is around minus 1.09. So what you can actually say is that the ecological system in microbes is actually obeying the May-Wigner criterion for stability. Okay, and here you see the colors are the different habitats again. All right? The final question we can answer is, now, is there a simple generative model for all these ecological networks we observe? We know that the internet, say, for instance, is scale-free, or maybe not. Then power grid networks in the US have a specific structure. Metabolic networks have a specific structure. And recently, we know that protein-protein interaction networks obey something that is called a sticky network topology. Okay? So what we do for this is basically we generate now a large collection of very simple random graphs. Many of them, compare them using our graphlet correlation distance, and see which of these models that all optimally fit to the individual networks we have, which model is best. And two particular interesting simple graphs are range-dependent graphs and sticky graphs, as I said, that are the standard models now for protein-protein interactions. Okay? And sticky just means that if I, in my network, have many neighbors and there is another guy having many neighbors, then the probability that we have a connection is the product of those. And microbial interaction networks are sticky. As you can see, here you see the average graphlet correlation distance to the different graph models, and lower means better. So across all the different habitats, we see that the sticky model and the range-dependent model, so both models for protein-protein interaction networks, are actually very good models. And here you can color those on the global map I showed you before. So the current hypothesis is that protein-protein interaction networks and microbial networks have a similar global topology. Okay, so that's my summary. Obvious thing is we need to validate all these interactions. And the best system so far, I would say, is the oral microbiome, because we know a lot about that. We can do image processing and can infer from image processing using classy fish, for instance, an interaction network and compare to what we inferred numerically. And if you're interested in these challenges and questions regarding the microbial ecology and statistics, we organize a workshop at the Simons Foundation, and I would love you to come. Okay. This my co-authors on the Speak Easy team, and thank you for your attention.
Very nice talk. I have a question regarding a topic you started with about these microbiome being like mixed with each other at some like level or like composed like together. And I was wondering if you have applied any of the deconvolution methods in order to separate them from each other before trying to like do further analysis. And by deconvolution, I mean like separating them like using a basis.
Well, what you do is basically, so you do this assay, you do amplification, and then you basically have all the 16S parts of the 16S gene. Mm-hmm. And then you assign this from a database, mm-hmm. you assign this to a specific group, mm-hmm. And because you don't have a spike in control, you basically can only measure relative abundances. You don't know the actual weight. So the basis data is these relative abundances, and that's all there is. And now you have, now I don't know how to apply convolution to that. You could obviously do grouping based on taxonomy. That is something we did, but this is the lowest level, so to say. This is on the species level.
So another question is, these barcodes, how stable they are? Are there mutations like in different, like individual samples, or?
Yes, so usually the cutoff to assign something to the same OTU is 97% sequence similarity, but this is active research. How to optimally, given these sequences, how to optimally assign them to, so to say, groups. This is absolutely still research, and there could be some example where it's unstable and others where it's not.
Thank you.
Okay, thank you.
I was intrigued by your statement at the end that you said that protein interactions and so protein interaction networks and microbial interaction networks have the same topology. Can you elaborate a bit on that? So what are, what numbers can you put on that? Are we talking about the key distribution or something else?
Well, we talk really about these actual generative models, right? So where we actually say, okay, so the sticky model and also I didn't explain the other model, the range-dependent one, but these are the standard models on a global scale that best fit protein-protein interaction network so far. Why and how this evolved, this is current ecology. We don't really know how ecological systems evolved.
Thank you.